channel of Clinical Care Insight. We have started a series of lecture on neuroanatomy and neurophysiology, okay? Until now, we already discussed about, you can say, many topics, fine. We have the initial, you can say, discussion started over on the basics about the neuroanatomy and neurophysiology. Basic, you can say, the outline of nervous system. Then we studied the fundamental structure and fundamental unit of nervous system, that's called as the neurons. We have this detailed study over the neurons. Then we studied, then we seen that, okay, apart from the neurons there are other cells, okay, which is playing a crucial role supporting these neurons. We term those as a supportive cell. Non-excitable cell are also called as the neuroglial cells, fine. Then we studied about the layers, okay. We seen the classification already in the overview of nervous system, and then we go ahead and in the further lectures we studied about the coverings that we referred as a meninges. We also seen the diseases, clinical correlations, fine. And then we studied about the one of the fluid, crucial fluid in the central nervous system, that's called as the, very good, it's a cerebrospinal fluid. Understood? No problem. Then we started a discussion over the synapse, okay, and we already covered about the synapse in detail, fine. And in today's lecture we will specifically talk about the neurotransmitters. Since from the previous lectures, synapse lectures, you come to understand that, okay, there are chemicals in the brain, okay, which helping in the transmission of impulse, okay, which is aiding in the transmission of you can say the impulses, fine. And we referred those chemicals as the neurotransmitters. Understood? So in these lectures we will understand first about the basic, what is the neurotransmitter, okay, how they are classified, fine. We will also see their discoveries, okay, which is, you can say, linked with one of the very amazing kind of experiment, and that is called as the heart experiment, fine, frog heart experiment. Understood? So here is your today's lectures outline. First we will deal with the introduction as we are doing, fine. But this introduction will be based on, because now you know that there are chemicals present in the central nervous system which is participating in the transmission of impulse, okay, transmission of action potentials. So there is no need to you can say go and define more about the neurotransmitter. Rather than we will see about the another chemicals which is also found in the nervous system, that is referred as a neuromodulators. Are you understanding? So basically we will establish one difference, because in the literature, medical literature, there is a very much confusion about the difference between the, you can say, they are used interchangeably, fine, they are used interchangeably, but we will see there are marked differences between the neurotransmitter and neuromodulators. So now let's study first the neuromodulator and the neurotransmitter, fine. We will also see, while after describing this, we will see about the criteria, that not the every chemical which is present in the, you can say, the central nervous system, they are referred as a neurotransmitter. No, there must be some criteria. If some chemicals, you can say, fulfilling those criteria, we will list down under the neurotransmitter, fine. Then we will see the experiment which I said, amazing experiment, the frog heart experiment, by which these neurotransmitters, they come into the, you can say, the literatures, medical literature, they come into the study, fine. Then we will see about the classification, that how these neurotransmitter, they are classified, fine. Then we will see how they are transported, how they are released, fine. And then we will see about the inactivation of neurotransmitter. Understood? No problem. So I am beginning today's, you can say, session by quoting Swami Vivekananda's one you can say the one quote. He is saying, we don't know how much energy is stored in us, okay, how much energy is stored in us, we don't know, fine. And the thing is, only, you can say, the minimal level of energy is known to us now, okay, but there are thousands of energy which is residing in us, fine. They are only just uncovered, fine. But due to our, you can say, the determination, due to our, you can say, passion, due to the, you can say, purity towards our karma, we can, you can say, once again channelize those energy. Understood? So no ever say that we are weak, fine. While we are considering that if we are weak, then we become weak. If we think we are strong, we become strong. So it's all about the thinking, fine. So let's start. I hope you all are fine. So first, what we are doing, we are writing the two terms. First one is called as the neurotransmitter. Neurotransmitter, fine. And in parallel, we will discuss about the neuromodulators. Neuromodulators. Now, as you know that both are chemicals. If we talk about their similarity first, then both are the chemicals, fine. But these chemical, okay, the neurotransmitters, they are involved in transmission of impulse, fine, the nerve impulse basically. Are you getting my points? Both are chemicals, okay, both are a kind of chemicals, isn't it? But the difference is, one is involved in the transmission of nerve impulse, where the another, they are not involved, you can say, directly in the nerve impulse, fine. They are not transmitting the nerve impulse, but they are involved, you can say, in monitoring, or another word you can say regulating, and it enhancing, fine, or you can also say it's amplifying the synaptic transmission. Are you getting my points? Yeah. The one that's called the neurotransmitters, they are actively participating, okay, in synaptic transmission. Probably you just remember from your previous classes, we've seen that there are nerves, fine. There are nerves. The first nerve we defined as the presynaptic neurons, postsynaptic neurons, okay, and there is a release of from here there is a release of neurotransmitter which bind with the postsynaptic membrane. Understood? So this is we refer as a synaptic transmission. This process we define as a synaptic transmission, fine. So in synaptic transmission, what is happening? There will be a, you can say, the transmission of action potential or nerve impulse or a message from presynaptic neuron to the postsynaptic neuron, okay. And in this you can say the chemicals, they are neurotransmitters, fine. You've seen the example of acetylcholine in detail in the synapse lecture. Understood? Now, then what are the role of these neuromodulators? The neuromodulators will enhance, or you can say increase, the frequency, fine, or they are amplifying, okay. They are involved in the amplification of transmission of this impulse. Understood? They are involved in the, you can say, transmission of impulse. Now, if we talk about chemically, then the another difference between the neurotransmitters and the neuromodulator is, this neurotransmitter, it can be amino acid, okay, chemically, it can be amines, fine, or it can be gaseous neurotransmitter, fine. There are the three natures. But if we talk about the you can say neuromodulators, they are only peptides. They are only the peptides, Fine. Now talk about the there you can say the storage. They are stored, you know it, there is a synaptic vesicles. Fine. Okay, they are stored in the small synaptic vesicles, small vesicles, or we can say vesicles. Fine, but they are stored in the large vesicles. See how many differences is, and we are just referring these are interchangeable. No, they are not interchangeable. The fundamental difference, you forget about these differences, but remember that they are involved, okay, in the nerve transmission, where they are not involved directly in the nerve transmission. Rather, they are, you can say, monitoring, regulating, fine, or enhancing, or we can say amplifying the synaptic transmission. Understood? No problem. Then if we talked about their chemical nature, these neurotransmitters, they are either amino acids, it can be amine, it can be the gaseous. Fine. Whereas if we talked about the chemical nature of neuromodulators, they are only peptides. They are only the peptides. Now about their, you can say, storage, they are stored in the small vesicles. We are referring those small vesicles as a synaptic vesicle. Fine, we are referring as a synaptic vesicles, isn't it? No problem. Now if we talked about their storage, they are stored in the large vesicles. Yeah, no problem. Are you understanding? Okay, fine. Now the another difference you can understood by their, you can say, production side. Generally the neurotransmitters are, you can say, formed, okay, they are formed in the soma or a cell body of a neuron. Okay, if this is the neuron, and you know it have the dendrites, fine, then these neurotransmitters, they are formed in the soma cell body, or they are transmitted through the axonema, or you can say the axon, and stored in the synaptic vesicle. Okay, but about the neuromodulators, since it is a, you can say, stored in a large vesicles, so you can just put your knowledge, your intelligence, and you understood, okay, it means they are not only, neuromodulators are not only, you can say, formed only in the soma or a cell body, it can form in anywhere in the neuron. Understood? Are you understanding? So if we talked about their production, the neurotransmitter production, they are mainly formed, okay, their formation in the soma or cell body, okay, and storage in the terminal end. Fine. And the storage of neurotransmitter is in the terminal end. Fine, terminal end of the neurons in the small vesicle that we are referring as a synaptic vesicles. Whereas about their formation, they are formed first of all anywhere in the neuron. Okay, so you can write down any place in the inside the neuron. Okay. Second thing about their storage, their storage, they are, okay, stored also anywhere. They are not restricted only to the synaptic, you can say the synaptic vesicle. Fine, no problem. Now the another difference we could you can say illustrate here is about that there may be a one kind of neurotransmitters is involved, okay, generally in the neurons, but now the research shows in the, you can say, one neuronal end there may be a different kind of neurotransmitters. Okay, but generally the neurons are, you can say, specific. There are GABAergic neuron, dopaminergic neuron, serotonergic neuron, fine. But this, you can say, the neuromodulators, okay, these and terminal end or anywhere where they are stored, okay, they are, you can say, they release one or more than one neuromodulators. Are you getting my points? Yeah, but generally if we talk about the neurons are generally specific. They release one kind of neurotransmitters. No problem? Understanding? So now you are not allowed to use the word neurotransmitters and neuromodulators. Remember, there are two different type of chemical present in our system. Okay, no problem. Once again, we are defining the neurotransmitter and neuromodulator. We said that generally the neurotransmitter and neuromodulators, they are, you can say, interchangeably used. Okay, they are used interchangeably. They are referring as they both are the same. But now we come to know that there are differences. Okay, the first difference is in their prime function. Their prime function is what? The neurotransmitter prime function is they are involved in the synaptic transmission, whereas neuromodulators, they are not, you can say, actively involved. Okay, fine. They are only monitoring. Fine. They are monitoring or regulating, or either they are enhancing or amplifying the synaptic transmission. Fine. The second difference is about their chemical nature. Both are chemicals, but both are of diverse kind of chemicals. The neurotransmitters, we will see one type of classification where we will classify the neurotransmitter on the basis of chemical. Fine. Now, the neurotransmitters can be amino acids, it can be amine, it can be gaseous, whereas, the neuromodulators are only peptides. They are restricted only to the, you can say, the peptic nature. Now, about their storage, they are stored in the small vesicles. They are stored in the small vesicles, whereas they are stored in the larger vesicles. Another difference is about their formation. They are formed, the neurotransmitter, they are formed in the, you can say, the soma or cell body. Okay, they are formed in the soma or cell body, and through the axon, through the axolemma, they are transported and come into the synaptic vesicle. They are stored in the synaptic vesicle. Fine. Whereas the neuromodulator is easy to understand, easy to remember, that they are in a large vesicle. It means their production would not only limited to the cell body. Okay, so in the neurons, inside the neurons, anywhere this can be synthesized. This can be synthesized. Another difference is neurons generally, I'm speaking generally, but you will come across there are neurons, okay, which secrete multiple kind of, you can say, the neurotransmitter, but generally the neurons will secrete the one kind of, store the one kind of neurotransmitter. Fine. But about the neuromodulators, they are one and more than one. They are one or more than one kind of neuromodulators. Fine, no problem. Now, once you understood about the neurotransmitter and neuromodulators, okay, if you want the example of the neuromodulator, then just write down some indocalfine. Okay, indocalfine. Okay, indocalfine as well as the endorphins. These are two kind of, I can say, the opioids. Okay, opioids, neuromodulators are divided, which we will see, you can say, after the break. But now just you understand that neuromodulators are also divided into the two categories. The one category is called as opioids. Fine. Another category is called as the non-opioids. Fine. These endorphins and indocalfine, fine, they are come into the opioid category. You know that opioids are generally used for what purpose? For sedation purpose, for relieving from the pain. It's used as an analgesic. That's why sometimes the, you can say, the dull pain, okay, which is, you can say, inhibited by this kind of chemicals, the neuromodulators. We won't feel the pain. We won't feel pain just because of this. It means our body also have one kind of, analgesics. They already have natural analgesic. Understood? So we have the natural analgesic. By the help of these chemicals, generally we will feel less pain. It's common observation. No, it's common observation. You've seen that if the two persons, they are injured by the same kind of, you can say, the trauma, the same strength of, you can say, the trauma, but both, they won't feel the same kind of, you can say, the pain. Why? The answer is, in there, you can say, the nerves, they are holding these neuromodulators, the endorphins and endocalfins, okay, which is releasing. Understood? That they are relieving from the pain. They are relieving from the pain. We will have the discussion over after the break about the neuromodulators, but now we will only focus on understanding the neurotransmitters. Understood? Okay, now we are going to define that. We said ki, okay, all the kind of chemicals which is present in our central nervous system, we won't say that all are the neurotransmitter. Then how will we understand that, okay, this chemical is neurotransmitter, this is not a neurotransmitter? There must be any criteria, okay, there must be any features which distinguish from the neurotransmitter from the rest of the chemicals. Okay, fine. So we are going to discuss those criteria. So write down the headings. Criteria for neurotransmitter. I am writing neurotransmitter as NTS short form. Okay. Now, what do you think? Give me any idea how we will, you can say, define this neurotransmitter. The one way to define the neurotransmitter is that about their production. You know that the neurotransmitters, they must, write it down, they must formed in the neuron. Are you getting my points? The first criteria is they must formed in the neuron. Understood? The second criteria is they must stored in vesicles, or we are more specifically the synaptic vesicles. Yeah. The another thing is they must bring the change in postsynaptic neuron. Are you getting my points? You know that these nerves, fine, these are the two neurons, okay, then the whatever is released from these synaptic vesicles, they must bring the change. When they bind with the their respective receptors, they must bring the change in this postsynaptic neurons. Otherwise we won't consider those chemicals as a synaptic, you can say the neurotransmitter. Are you getting my points? Yeah. And the another criteria is after the, you can say the, after executing the effect, okay, they must become inactive. Now what it means? It means okay, this much receptor is present and now all the receptor is occupied, then whatever the left, you can say the neurotransmitter is present in this gap that we referred as a synaptic cleft, they must be inactive. And inactive by what? The one thing is there are the, you can say the natural enzymes present over here, okay, which can break down and that are reabsorbed again in the presynaptic neuron. Are you getting my points? Understood? No problem. Now, is there any other way to you can say criticize these neurons, these neurotransmitters? Yeah? No? Okay. So these are the some of the criteria for, you can say, defining the neurotransmitters. Now, once we understood, let's summarize it once again. First is it must form in the neuron. Okay, now it must stored in the synaptic vesicles. When it's you can say released from these synaptic vesicles, fine, they must bring some biological change in the postsynaptic neuron. Are you getting my points? So they must bring the biological change in the postsynaptic neuron. The last thing is when they are executed, when their duty is over, they must be become inactive. They must become inactive. So these are the four criteria by which we can make one kind